Ready, madam?
Ready, sir. Shall I start?
Start, madam.
Good morning, students. This is Sarala, working as JL at Hasanparthy, Warangal Urban. Welcome to webinar revision classes. Today, I have discussed about the unit biotechnology. This biotechnology unit is very, very important for IP as well as NEET and MSET point of view. This unit in IP, this biotechnology unit weightage is 20 marks. One long answer as well as two very short and two short questions are coming from this unit biotechnology. Along with the IP, it is also most important for NEET and MSET. Okay, today we have to discuss about the long answers in this unit biotechnology. Let us discuss about the first long answer. Give a brief account of the tools of rDNA technology or recombinant DNA technology. So, two long answers are very, very important. First long answer is tools of the recombinant DNA technology or rDNA technology. Before this question, first I have explained about rDNA. rDNA, recombinant DNA. Recombinant DNA means first we have to take the desirable genes from the host organism. So this DNA molecule which consists the desirable genes, and these desirable genes are isolated from this host, and we have to take the small piece of DNA from this host organism, and then this desirable DNA is incorporated into a vector. In most of the cases, plasmids are used as vectors, and plasmids are present in bacterial cell. It is the unique feature of bacterial cell. Plasmid is the unique feature of bacterial cell. Plasmid, which is a single-stranded, plasmid is the double-stranded DNA molecule, self-replicating double-stranded DNA molecule, which is present in the bacterial cell. So this desirable DNA is incorporated into the plasmid. So this desirable DNA molecule is inserted into the plasmid, and this is called as rDNA or recombinant DNA. So this recombinant DNA is inserted into a suitable host. That organism is called as genetically modified organism. Genetically modified organism means its genetic material DNA is modified or altered, and desirable genes are present in that host. Okay, it is called as rDNA or recombinant DNA. Okay, so the major tools of rDNA, recombinant DNA technology is first tool is restriction enzymes, and second tool polymerase enzymes, third one ligases, fourth one vectors, and fifth is the host organism or the competency or acceptancy of the host organism. Once again, I repeat it. The major tools of rDNA technology are restriction enzymes, polymerase enzyme, ligases, vector, host organism or competency or acceptancy of the host organism. Okay, first we have to discuss about the first tool, restriction enzymes. So restriction enzyme. Restriction means they have to cut the DNA molecule at specific location. This restriction enzymes also called as molecular scissors of the biotechnology. They have to cut the DNA at a specific unique point. Okay. These two enzymes responsible for restricting the growth of bacteriophages. Bacteriophage means virus which can infect the bacteria. It is called as bacteriophage. Bacteriophage nothing but virus, which infects the bacteria. That viruses are called as bacteriophages. The two enzymes, they are responsible for the restricting the growth of bacteriophage in E. coli bacteria. Those are isolated from this E. coli, Escherichia coli bacteria. And out of these two enzymes, one of these enzymes added methyl groups to the DNA molecule, and the other enzyme cut the DNA at specific location. Later, these enzymes are called as restriction endonucleases. This restriction enzymes also called as restriction endonucleases. First two enzymes are isolated from this Escherichia coli bacteria. One of these enzyme adds methyl group to the DNA molecule, and other enzyme, they cut the DNA at a specific unique location. These enzymes are called as restriction endonucleases. They are also called as molecular scissors of the biotechnology. So one of the restriction enzyme is Hind2. Okay, Hind2 always cut the DNA molecule at a particular point by recognizing a specific sequences of 6 base pairs. Each restriction enzyme having a specific recognition site. The recognition site is always varies from one enzyme to another enzyme. This Hind2, it is one of the type of restriction enzyme which always cut the DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as the recognition sequence for Hind2. The recognition sequence or site which is differ from one enzyme to another enzyme. Today, we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria. These restriction enzymes are present in bacterial members only. Bacteria, they are unicellular prokaryotic organism. They having this restriction enzymes. They also having the plasmids. Plasmids are used as vectors. So another restriction enzyme is EcoR1. EcoR1 means which is isolated from Escherichia coli bacteria. This EcoR1 comes from Escherichia coli strain RY. Its number is 13. In EcoR1, the letter R is derived from the name of 2015 and 16 196 biology strain, R as well as Y, and 13. It is the number as well as sequence which is present in the E. coli bacteria. When we observe the naming of the enzyme EcoR1, which is the restriction endonuclease enzyme. So this, when we observe this EcoR1, Escherichia coli, first letter is the genus name. Second two letters indicates the species name. R is the series of the strain, and the number is the serial number which is present in the strain, which is derived from the name of the year 2015 and 16 196 biology strain. And the Roman numbers following the names indicates the order in which the enzyme were isolated from the strain of the bacteria. This one Roman letter, Roman numbers following the names indicates the order in which the enzymes were isolated from that strain of the bacteria. Larger class of enzymes called as nucleases. Nucleases means they are present inside the nucleus of the cell. These are two kinds of nucleases, exonucleases as well as endonucleases. Exonucleases as well as endonucleases. Exonucleases removes the nucleotides from the ends of the DNA molecule. Nucleotide means nitrogen base, sugar as well as phosphate radical is considered as nucleotide. nucleotide. Nucleotides are present in both DNA as well as RNA. Nitrogen bases are two types, purines as well as pyrimidines. Purines are adenine and guanine, pyrimidines are thymine and cytosine. So in the case of DNA, thymine is present. In the case of RNA, instead of thymine, uracil is present. Nucleotide means nitrogen base plus sugar plus phosphate ion is considered as nucleotide. This exonucleases, they have to remove the nucleotides from the ends of the DNA molecule. Whereas endonucleases, they can cut the DNA at specific point. It is the difference in between the exonucleases and endonucleases. Exonucleases remove the nucleotides. Endonucleases cut the DNA molecule at specific location. For example, which is the DNA molecule. DNA is the double-stranded molecule. One strand is runs in 3' to 5' direction. Another strand is runs in 5' to 3' direction. And this endonuclease, which cut the DNA at a specific location. It is the major function of endonucleases. Endonucleases, they can cut the DNA molecule at a specific position. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA molecule. Once again, I repeat this point. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA molecule. Palindromic sequence means when we observe a letter, for example, madam. So when we read this word, madam, in any direction, from this direction as well as from this direction, same letters are present. That sequence is called as palindromic sequence. So these restriction enzymes, they can recognize only palindromic sequences which are present in the DNA molecule. So it is the diagrammatic representation of action of restriction enzyme. Which is the DNA which is present in a particular organism with desirable genes. And it is the vector DNA, vector which carries the foreign DNA from one organism to another organism. It is called as vector. So plasmids, plasmids, phasmids, as well as ZIAP. ZIAP means yeast artificial chromosome. They are used as vectors. But in most of the cases, plasmids are used as vector. Vector means which carries the foreign DNA from one organism to another organism. So it is the foreign DNA molecule, foreign DNA, which is isolated from a organism with desirable genes. Desirable genes means it having a unique character. It is either a disease resistance capacity or high yielding capacity or insect resistant, pest resistant capacity. So this EcoR1 is the one of the type of restriction enzyme which cuts the DNA between the bases G and A only. G and A means which is the recognition sequence for EcoR1 enzyme. EcoR1 is the restriction enzyme which cuts the DNA in between G and A, guanine and adenine only. When the sequence is present, G, A, A, T, T, C, guanine, adenine, thymine, and cytosine, which is present in the foreign DNA. So, which cut the foreign DNA in between these palindromic, in between these sequences. Okay, it is the vector DNA. So, the same enzyme, EcoR1, which cut the vector DNA at the same position in between G and C. So from G and C, it cut the foreign DNA between G and A sequence. So it is the vector DNA which is the foreign DNA. So by the action of restriction enzymes, they can cut the vector DNA as well as foreign DNA, and they can form a specific ends called as sticky ends. So this two ends, they are fit within each other. So DNA fragments join at sticky ends. This foreign DNA is easily inserted in the vector DNA by the formation of sticky ends. So this foreign DNA is inserted into the vector DNA and they can form a recombinant DNA or rDNA. So which is the action of the restriction enzymes. Palindromic DNA is a sequence of base pairs that read palindromes. These are a group of letters that form the same words when read both forward as well as backward direction. Example, Malayalam or Madam or Liril. These are the palindromic sequences. Palindromic sequences means the two strands having the same base pairs. So it is these are the two strands of the DNA molecule. One strand is runs in 5' to 3' direction. Another strand runs in 3' to 5' direction. Both these strands are complementary with each other. So in this strand, the nitrogen bases are present in GAATTC. In the another strand, CTTAAG. So GAATTC, CTTAAG. So both the strands having the same bases which are runs in opposite manner. These are called as palindromic sequences. Restriction endonucleases, they can always cut the DNA at this palindromic site. This palindromic sequence is also differ from one restriction enzyme to another restriction enzyme. Restriction endonucleases are used in genetic engineering or recombinant DNA technology to form recombinant molecules of DNA, which are composed of DNA from two different sources or genomes. Listen here. Restriction endonucleases are used in genetic engineering. Genetic engineering, nothing but rDNA technology, recombinant DNA technology to form recombinant molecules of DNA, which are composed of DNA from different sources. One DNA source, first source is from foreign organism with desirable genes, and another one is host organism. DNA is composed from two different sources. It is called as rDNA. So it is the plasmid. It is the foreign DNA with desirable genes. This foreign DNA is inserted into the plasmid or vector and it is called as rDNA. And this rDNA, recombinant DNA, is inserted into a suitable host and it is considered as rDNA or recombinant DNA, which are composed of DNA from different sources or genomes. These DNA fragments have the same kind of sticky ends and this can be joined together using DNA ligases. Ligases are the adhesive. One of the type of enzymes, they are adhesive in nature. They can join the foreign DNA with vector DNA. It is a diagrammatic representation of rDNA technology, which is the foreign DNA, and it is the vector DNA, nothing but plasmid. This foreign DNA is cut into small fragments with the help of restriction endonuclease, and this vector DNA is also cut at a specific location with the help of same restriction enzyme, and these two are joined with each other with the help of ligase enzyme, and it is called as rDNA or recombinant DNA, which is inserted into a suitable host, and this host undergo replication, and it can multiply the copies of desirable DNA molecules. Next, coming to the second tool, polymerase enzymes. First tool is the restriction enzyme. It is completed. Then coming to the second tool, polymerase enzyme. Polymerases. Polymerase is an enzyme that synthesizes long chains of polymerase or nucleic acids. Polymerase enzyme, they can play an important role in rDNA technology or recombinant DNA technology. It is used to synthesize the long chains of polymerase or nucleic acids or long chains of polymers of nucleic acids. DNA polymerase is used to assemble DNA molecule respectively by copying a DNA template strand using base pair interactions. With the help of this DNA polymerase enzymes, the DNA molecule is copying at a specific point. In this rDNA technology or recombinant DNA technology, tagged DNA polymerase enzyme is used in PCR, polymerase chain reaction, to amplify the desirable gene. Tagged DNA polymerase enzyme, which is isolated from bacteria Thermus aquaticus. This tagged DNA polymerase is isolated from tagged DNA polymerase enzyme is isolated from a bacterium Thermus aquaticus, which is used in rDNA technology for amplifying the DNA molecule. Next, third tool is ligase enzyme. Ligase is an enzyme that links two different DNA molecules, that is foreign DNA molecule as well as vector DNA molecule. These ligases are adhesive in nature, which are used to join the two different DNA molecules. In DNA cloning, DNA ligases are used to join the alien DNA with plasmid DNA, which is used to join the alien DNA, nothing but foreign DNA, with plasmid DNA, which is the third tool, ligases. Next, coming to the fourth tool, that is cloning vectors. Vector means which carries the foreign DNA from one organism to another organism. Once again, I repeat the definition of vector. Vector means which carries the foreign DNA from one organism to another organism. So in this rDNA technology, plasmids, bacteriophages have the ability to replicate within the bacterial cells independent of the control of chromosomal DNA. Plasmids, bacteriophages, plasmids, are used as vectors, and they are having the ability to replicate within the bacterial cell independent of the control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. the copy number of this bacteriophage as well as the copy number of the plasmid is varies from one bacterial to another bacterial cell. Some plasmids may have only one or two copies per cell, whereas other may have 15 to 100 copies per cell. The number of plasmids which is present in the bacterial cell is called as copy number. This copy number is varies from one bacterial cell to another bacterial cell. So when we observe the structure of plasmid, so this PBR 322, it is the one of the widely used plasmid in the rDNA technology, which is artificially constructed plasmid. PBR, PUC are widely used plasmids in the rDNA technology. It is an artificial plasmid. PBR, P stands for plasmid, B Bolivar, R stands for Rodriguez, 322 is the number, the sequence of the plasmid. P plasmid, B Bolivar, R Rodriguez. Bolivar and Rodriguez, these two scientists, they are constructed this plasmid. 322 is the serial number of the plasmid. So when we observe the structure of this plasmid, so the first unique point of the plasmid is oocyte. Oocyte means origin of replication. So this oocyte is also differ from one plasmid to another plasmid. The first unique feature of the plasmid is oocyte, origin of replication, which is takes place at a unique point. And the second unique point of the plasmid is recognition sequence. The recognition sequence is also differ from one plasmid to another plasmid. So this AMPR as well as TET-R. AMPR means ampicillin resistant region. TET-R means tetracycline resistant region. These ampicillin resistant region as well as tetracycline resistant region, it is used as antibiotic resistant regions. By the presence of this antibiotic resistant regions, they have to tolerate some antibiotics which are added to the culture medium to prevent the contamination. And they also use it as marker genes for the identification of the rDNA from normal DNA molecule. And this ECHO R1, CAL1, and HIND3 and BAMH1, SAL1, these are the restriction endonuclease enzyme sites. ECHO R1, CLA1, HIND3, BAMH1, SAL1. These are the restriction endonucleases for this specific plasmid, and this restriction enzymes, they have to cut the plasmid at this specific sequence only. These are the unique features of the plasmid. First feature is oocyte, origin of replication. And the second feature is selectable marker genes. Selectable marker genes, tetracycline resistant region as well as ampicillin resistant gene. Antibiotic resistant regions are considered as selectable marker region. Next, cloning site. Cloning site is also specific for the plasmid. And vectors which are most commonly used in the plants and animals. The first common vector is TI plasmid, tumor-inducing plasmid, which is present in the Agrobacterium tumefaciens. TI plasmid, tumor-inducing plasmid, which is present in Agrobacterium tumefaciens bacteria, which is used in the plants, so modifying as a cloning vector. In the case of animals, retroviruses are most commonly used as vectors in the case of animal cells. In the case of plant cells, TI plasmid is used as vector. In the case of animal cells, retroviruses are used as vectors. Retroviruses, its pathogenicity or pathogenicity is suppressed and it is used as vectors in the case of animal cells in the gene transfer mechanism. Next, last step in the last tool or last step in the DNA technology is competency of the host cell. So in this competency, nothing but acceptancy of the host cell. So it is the host molecule and it is the foreign DNA along with the bacteriophage, which is integrated or which is transferred into a host DNA. First, the host organism is ready to accept this foreign DNA. The acceptancy or the competency of the host organism is necessary in this rDNA technology. If that host organism is ready to accept the foreign DNA, then that foreign DNA is easily inserted into the host DNA with the help of biolistic or gene gun method. It is the last tool which is in the rDNA technology. Majorly 5 tools are present in the rDNA technology. First tool is the restriction enzymes. Second tool is polymerase enzyme. Third tool is ligase enzyme. And fourth tool is vector. And last but not the least, competency or acceptancy of the host organism is the last tool. So it is about the complete explanation of the tools of rDNA technology or recombinant DNA technology. Next, coming to another long question, it is also very, very important for IP. That is, explain the process of recombinant DNA technology. Process or steps of the recombinant DNA technology. So when we observe the process, these are the major steps which is involved in the rDNA technology. The first step is isolation of genetic material, nothing but DNA. And the second step is cutting of the DNA at a specific location. And third step, separation and isolation of DNA fragments. Fourth step is insertion of isolated gene into a suitable vector. And the fifth step is insertion of rDNA into the host cell. So these are the 5 major steps which are present in the process of rDNA technology or recombinant DNA technology. First step is isolation of genetic material DNA. Second step is cutting of the isolated DNA at specific location. Next, third step is separation and isolation of DNA fragments. And fourth step is insertion of isolated genes into a suitable vector. And the last step is insertion of rDNA into a host cell. Let us discuss about the first step, isolation of genetic material DNA. This genetic material DNA is present inside the nucleus in both plant cells as well as animal cells, which is present inside the nucleus. In the case of prokaryotic cell, nucleus is poorly developed. At the place of nucleus, only genetic material DNA is present. Okay, so this isolation is achieved by treating the bacterial cell or plant cell or animal cell with enzymes such as Lysozyme in the case of bacteria, Cellulase in the case of plant cell, Chitinase in the case of fungal cell. The enzyme which is used for the digestion of cell, which is depend on the type of the cell. It is the bacterial cell, the enzyme is Lysozyme. It is the plant cell, Cellulase enzyme is used for the digestion of cell wall. In the case of fungal cell, Chitinase enzyme is used for the digestion of cell wall. You know that Genes are located on the long molecules of DNA intertwined with the proteins such as histones. Histone proteins are present in the both plant cell as well as animal cells. In the case of bacterial cells, histone proteins are absent. Okay, in the both plant as animal cells, DNA is intertwined with histone proteins. And along with these proteins, RNA is also present along with DNA. RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by the treatment with protease. Along with DNA molecule, proteins as well as RNA molecule is present. So that's why proteins are removed with the help of protease enzyme. RNA molecule is removed with the help of ribonuclease enzyme. So with the help of this enzyme action, cell wall as well as proteins as well as RNA can be removed and DNA molecule is separated, and the DNA is isolated or separated with the help of a glass rod. That process is called as spooling. Glass equipment is present. With the help of that glass equipment, DNA molecule is or DNA threads are removed with the help of a phenomenon is called as spooling. Next, second step is cutting of DNA at a specific location. DNA molecule, by the action of restriction enzyme digestion, DNA is cut at a specific location. So restriction enzyme digestion are performed by incubating the purified DNA molecule with the restriction enzyme. For example, it is the isolated DNA fragment which is present in the form of small fine threads. It is a container. So in this container, restriction enzyme is added to this DNA molecule, and this restriction enzyme cut the DNA at a specific unique point, and this process is called as restriction enzyme digestion. Restriction enzyme digestion means restriction enzymes are added to the purified DNA molecule which cut the DNA molecule at a specific unique location, and that step is called as restriction enzyme digestion. After having cut the source DNA as well as the vector DNA is also cut at a specific site with the same restriction enzyme. Listen carefully. Source DNA is cut at a unique site with the help of a restriction enzyme, and the same restriction enzyme is also used to cut the vector DNA at a specific site. And after having the cut the source DNA as well as the vector DNA with a specific restriction enzyme, then the cut out the gene of interest from the source DNA. Gene of interest nothing but desirable genes. From the source DNA, the cut vector with a space are mixed, and with the help of ligase enzyme is added. After the restriction enzyme digestion, then ligase enzyme is added to ligase the both specific restriction DNA as well as specific vector. Both are ligated with the enzyme ligase. This results in the formation of rDNA or recombinant DNA. So when we observe this diagram, so it is the echo R1 restriction endonuclease which cut the host DNA at a specific site, and which cut the foreign DNA, not host DNA, foreign DNA at a specific unique site, and the same DNA which is used to cut the plasmid. which is the plasmid, and the same echo R1 enzyme which is used to cut the plasmid, its same recognition sequence. And this plasmid DNA as well as foreign DNA both are inserted or joined with each other with the help of ligase enzyme, and it is called as rDNA. So this is the Hind3 enzyme which cut the DNA at a specific point. Hind3 is also one of the restriction enzyme, and the recognition sequence is specific for one enzyme to another enzyme which cut the DNA at this specific unit. Next, third step is separation and isolation of DNA fragments. These fragments can be separated by a technique known as gel electrophoresis, agarose gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards anode under an electric field through a medium or matrix. That medium is nothing but agarose gel. DNA molecules are negatively charged, and they can be separated according to their size under an electric anode through a medium or matrix. So it is the agarose gel electrophoresis. So these are the gel. It is the gel, and these are the DNA bands. These DNA bands are moved on this medium or matrix that is nothing but agar gel, and these are the electrodes, and these DNA fragments are separated according to their size. This gel or matrix, agarose gel electrophoresis, is the most suitable method for the separation of DNA molecules according to their size. Nowadays, the most commonly used matrix is agarose, which is a natural polymer which is extracted from seaweeds, nothing but red algae, rhodophyceae members. Agarose gel, which is extracted from red algae, rhodophyceae members. The DNA fragments separated according to their size through sieving effect provided by the agarose gel. You can see bright orange colored bands of DNA in a ethidium bromide stained gel. Ethidium bromide gel is used for the identification of DNA fragments. This ethidium bromide stained gel exposed to UV light, the separated bands of DNA are cut out from the agarose gel and are extracted from the gel piece. This step is known as elution. Elution means DNA fragments are easily identified with the help of ethidium bromide stain. When UV light is exposure on the DNA fragments by the presence of this ethidium bromide stained gel, DNA fragments are appear orange color, and those orange colored DNA fragments are separated or extracted from the gel. This step is known as elution. The recombinant DNA by joining them with cloning vectors. So next, coming to the fourth step, insertion of isolated gene into a suitable vector. To isolate a plasmid from the bacterial cell subjected to the centrifugation to separate the plasmid. Plasmids are present inside the bacterial cell. So that isolated desirable genes are inserted into a vector. These vectors, plasmids are used as vector. These plasmids are separated from the bacterial cell, and that bacterial cell is subjected to the centrifugation to separate the plasmid, and this plasmid is cut the same restriction enzyme action. So when the foreign DNA is cut with the help of a echo R1. The same echo R1 is used to cut the plasmid DNA, and the source DNA as vector DNA are mixed together with the help of DNA ligase enzyme. This results in the formation of recombinant DNA or rDNA or chimeric DNA. So it is the diagrammatic representation of rDNA or recombinant DNA, which is the foreign DNA with desirable genes. Desirable genes which is either a disease resistance capacity or stress resistance capacity or high yielding capacity. Okay, which is present in the foreign DNA, which is the vector DNA, that is nothing but plasmid DNA. So this foreign DNA as well as vector DNA is cut with the help of same restriction endonuclease enzyme. It is either EcoR1 or BAMH1 or HIND3. So these two DNA sources are cut into a specific recognition sequence with the help of restriction enzyme, and this foreign DNA is joined with this vector DNA with the help of ligase enzyme. Ligases are adhesive in nature. They have to join the foreign DNA to plasmid DNA. It is called as rDNA or recombinant DNA or chimeric DNA, which is transferred into a suitable host. Okay, it is the diagrammatic representation of the process of rDNA technology. Next, amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. Amplification of gene of interest means amplification nothing but multiplication of desirable gene. So in this reaction, multiple copies of gene or DNA of interest, interested gene, nothing but gene with desirable genes are multiplied by the using of a technique known as PCR, polymerase chain reaction, in vitro using two sets of primers. Primer, it is a small chemically synthesized oligonucleotide that are complementary to the region of DNA, and the enzyme is DNA polymerase enzyme, especially tag DNA polymerase, TAQ, tag DNA polymerase enzyme is used in this PCR. And a primer, oligonucleotide primer, along with this tag DNA polymerase enzyme is used in this PCR technique. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified approximately billion times. Billion copies are made within fraction of seconds with the help of PCR technique. The amplified fragment is desired can now be used to ligate with a vector for further cloning. So it is the diagrammatic representation of PCR. So polymerase chain reaction. First step is denaturing. Denaturing means two strands. DNA molecule in general, which is present in double helical structure. These two strands are separated. That process is called as denaturing, which is takes place at high temperature, up to 95 degrees Celsius temperature. So that's why at high temperature, normal DNA polymerase enzyme is not tolerated. So that's why tag DNA polymerase enzyme is used in this PCR technique. Okay, and in this second step is annealing. Annealing means oligonucleotide primer is used to polymerize the DNA molecule. And the third step is extension. Extension means the desirable DNA segment or desirable genes are extended through a polynucleotide chain with the help of oligonucleotide primer with the help of tag DNA polymerase enzyme. It is the PCR cycle, polymerase chain reaction, which is important for short answer. Okay, in this biotechnology unit, in this long answer, this PCR, it is amplification of gene with the help of PCR, which is a short question. It is important. Next, last step is insertion of recombinant DNA into a host cell or host organism. Once rDNA is ready, which is inserted into a suitable host cell or host organism. This insertion takes place with the help of several methods. If a recombinant DNA bearing gene for resistant to antibiotic, that is ampicillin, which is transferred into the E. coli cells, the host cells become transferred into ampicillin resistant cells. The transformed cells or transformants are identified from non-transformants with the help of ampicillin resistant genes. If the transformants having ampicillin resistant genes and the non-transformants that does not having ampicillin resistant genes. So this ampicillin resistant gene, which is used as marker gene to identify the transformants from non-transformants which are transferred into the recipient cells. Insertion of rDNA into a host cell. This is the rDNA, recombinant DNA, which is inserted into the host cell. So with the help of gene gun method or biolistic method or micro-injection method, several methods are present in the gene transfer method. They are micro-injection method, gene gun method, or biolistic method. Any one of the method is used to transfer this rDNA into the host cell, and the host cell become transformed cell. Okay, it is the insertion of rDNA into the host cell. Next step is obtaining the foreign gene product. DNA gets multiplied in the host cell in almost all recombinant technologies. The ultimate aim is to produce the desirable product. It is either a protein or enzyme or antibiotic, or it is either a hormone like insulin. The main aim of the rDNA technology is finally to get a desirable product. The target protein, one has to consider producing it on a large scale. It is either a protein or a hormone or an enzyme. It is produced on a large scale within the short period. It is called as recombinant protein or recombinant enzyme or recombinant hormone. So the cultures may be used for extracting a desired protein. The development of bioreactor. Bioreactor is a large vessel which is used to bioreactor, which is a large vessel which is used to obtain the product in the large scale within the short period. These bioreactors can be thought as a vessel in which raw materials are biologically converted into a specific product, individually an enzyme, using a microbial, plant or animal or human cells. A bioreactor provides the optimal conditions for achieving the desirable products by providing optimum growth condition. Optimum conditions means suitable temperature, suitable pH, and salt, and by the addition of essential vitamins. And if that organism is aerobic organism, oxygen is supplied to the bioreactor. If that organism is anaerobic organism, oxygen supply is prevented, which is this oxygen supply is depends on the nature of that organism, which is either aerobic organism, oxygen supply is necessary. If that organism is anaerobic organism, oxygen supply is prevented. Okay, this bioreactor which provides the optimal conditions for the growth of that particular host organism to get a desirable product within the short period in the large scale. The last step is downstream processing. Downstream processing, it is important for very short answer. Downstream processing means the process include separation and purification, which are collectively referred to as downstreaming process. Downstream process means separation and purification of desirable product. That is either a protein or hormone or enzyme, which is separated from the bioreactor, which is undergo purification, and that process is called as downstream processing. The product has to be formulated with suitable preservative. The purified product is formulated with suitable preservative. Such formulation has to undergo thoroughly clinical trials in the case of drugs. Number of antibiotics, number of vaccines are prepared with the help of rDNA technology or biotechnology. So that antibiotics as well as vaccines are not directly released into the market. First, clinical trials are conducted in the case of antibiotics as well as vaccines. Clinical trials are compulsory. How that vaccine are performed, how that antibiotic is performed, its action on lower organism, its function is very well, then it is released into the market. So clinical trials are compulsory in that product. Strict quality control testing for each product is also required. The downstream processing and the quality control testing vary from product to product. The downstream processing and quality control as well as quality control testing is differ from one product to another product, which is differ from enzyme to protein, protein to hormone, hormone to an organic acid. That purification, isolation, purification and quality control and testing is differ from one product to other product. So it is about the total process or steps of rDNA technology. The two long answers, both are very, very important for IP purpose. From these two questions, any one of the question is coming in IP exam.
Hello, madam.